All right, thank you, Celeste. First of all, I just want to say it really, truly is an honor to be invited to speak here at something that's dedicated to Larry. He had so many great students. It's an honor to feel like you're part of that community. So I thank you for inviting me. It's a real pleasure to be here. Those of you who have been around for a while may remember when the genetics department was in the J Wing, and you probably remember the genetics department seminar room, and you probably remember that across the hall from the seminar room was a little conference room where before every seminar, coffee and cookies were served. That's where I first met Larry. So when I came to the genetics department as a student, Larry was on sabbatical. That was in the era before departmental websites, before you could see faculty photos of everyone. So I had no idea who Larry was. I knew there was some guy, because of a 2-sentence description in the Peterson Guide, I knew there was some guy who worked on something called Drosophila. And it's like, why the hell would you work on an insect? You know, this was 1980. This was just a few years after the discovery of restriction enzymes, the development of molecular cloning, and I thought cloning is cool, and that's what I want to do. And, you know, a genetics department, that would probably be a great place to go to really go down that path and do some interesting things. And so I came here, and in that first year, Larry was on sabbatical. He had no students. He'd graduated everyone before he went on sabbatical. He wasn't around. I didn't know who he was. We had never seen him. He really didn't have any part of departmental life as I knew it. He just wasn't part of anything. And of course, all of you realize that was a very mistaken impression of the genetics department. So anyway, back to meeting Larry. So as a very early second-year graduate student, I went into the conference room across from the seminar room to get coffee and cookies before the seminar. What graduate student turns down free cookies? None that I know. And so I went in there, and the departmental staff had not brought the cookies in yet. And I was the only one there. And so I just stood there and waited, and I thought, I know they're going to show up pretty soon. And I was in there all alone, and pretty soon a large mustachioed gentleman walked in, and he paused, and he looked around the room, and he looked at me, and he said, "Why don't you go get the cookies?" And it did not feel like a request. So I did it. I went and got the cookies, and but I remembered that moment for a long time that day and ever since. But that was when I first met Larry. So when I started graduate school in the genetics department, working towards a PhD in genetics, I had never taken a formal genetics class. And, you know, I realized that during my career there have been several extremely fortunate moments for me. I benefited from a lot of good fortune. One of them was to end up in this department. And even though I knew nothing about genetics really, and I think that was due to Herschel Roman, who I'm told thought I was a farm boy from the Midwest, and Herschel, being a farm boy from the Midwest himself, thought I could be a good addition to the department. So I wasn't a farm boy, but I'm thankful for that misunderstanding. So the first year, having no genetics training, was actually, it was really hard for me. It was very difficult. But the second year was inspirational. And that was entirely due to Larry and his class on sort of classical genetics. And that's where I really learned what genetics was all about. So I want to share a quote from Moeller, not Robert, that I think sort of gets at appreciating what Larry taught me, and it's this. So this is from an address Moeller gave in 1926, And he says it's commonly, well, first of all, he's talking about how people might enter the field of biology and become biologists in contrast to really being a geneticist. And he says it's commonly felt that the picture of living things painted by the modern geneticist, 1926, is just too good to be true, or shall we say too true to be good. I love that twist of a phrase. And that living things just cannot, and in any way do not, work with such precision and automaticity as he claims, and that even if they should, it would not be in good taste to have anything to do with them. So this is just it's a wonderful paragraph or two at the beginning of this paper where Muller invents words like automaticity, and he also invents another word, which the quote is too long to put all here, but he calls biologists in general obscurantists. But it's this that I think sort of gets to the appreciation that I have for genetics that was inspired by Larry, and that is that the precision, the reproducibility of genetics allows you to do carefully designed experiments, look at numbers and believe them and trust them to tell you things about how chromosomes behave when they are completely invisible. So that's what I loved when I took Larry's class. That's how I learned to love genetics, and that's what I owe to Larry, among many other things. All right, so in sort of thinking about the theme of today, I am not going to talk about our most recent work. Instead, what I'm going to do is I've picked out two items to talk about that I think are experiments that Larry would have appreciated. So that's why I've chosen these. They're experiments that we've done a while back. And I'm also going to break one other rule that I have. So I live in a biology department. I have to pretend to be a biologist, even though I don't really think of myself as a biologist. And living in a biology department and giving seminars to biologists, I've learned that giving seminars about genetics is sort of like Fight Club. So if you're going to give a seminar about genetics, don't talk about genetics. So I'm going to break that rule, and I'm going to talk about genetics. And I hope it's in a way that Larry would have appreciated. So the first thing that I want to talk about are experiments that talk about segregation of genotypes in mitosis. And that's a little bit of an odd title because we think of mitosis as preserving genotypes, right? You don't segregate genotypes. The whole point of mitosis is to faithfully transmit genetic information from one cell to another. But you can segregate genotypes if you have recombination. And this is a phenomenon that the basis of which was elucidated by Kurt Stern in 1936. And basically, the phenomena is that occasionally, if you have a heterozygote, let's say big B over little b, you can have a crossover, not a meiotic crossover, but a crossover in mitosis in G2 of the cell cycle. So this is between two non-sister chromatids. And that exchange then will allow these chromosomes to segregate in different ways that Kurt Stern called X segregation. And X segregation is when the centromeres attached to the two chromatids that recombine segregate away from each other. So I've just numbered these 1, 2, 3, 4. When 2 and 3 centromeres segregate away from each other, that's X segregation. And that kind of segregation produces homozygous daughter cells. Okay, so that allows you to see a mosaic patch of cells or a patch of cells in an animal that is genetically different from what the genotype of that animal was initially. Now, the alternative is Z segregation, where these two chromatids segregate together. And although this cell has recombinant chromosomes, it doesn't change the genotype of the daughter cells. So Kurt Stern thought these are probably equally frequent. If you get X segregation, you can do useful things with that because you can study mosaics, learn things about development. Around the time that actually I finished graduate school, Sergio Pimpinelli and Pedro Ripoll had a very beautiful paper where they took a cytogenetic approach to determining if that assumption, X and Z are equally frequent, if that assumption was in fact reality. And the answer was it wasn't, that in fact X segregation outnumbered Z segregation by at least 2 to 1 after they induced exchange with X-rays. Now, there are still some remaining questions because, first of all, we don't know how much recombination occurs in G1, and secondly, we don't know what the basis for that preferential X segregation might be. So when I discovered that you could use site-specific recombination to induce exchange between homologous chromosomes, that gave us a new opportunity to go back and re-examine this question. And now, because this exchange is so very efficient and so very frequent, it made it pretty easy to set up experiments where we could answer those other two questions. So here's how the first half of the experiment goes. The first part is that we have to have a way to detect when recombination occurs. And the way we did that is, first of all, we have these flip recombination target sites. So the flip recombinase is going to catalyze recombination between those if we induce flip expression. And we set these up so that on one side, we've got the coding sequence of the white gene, which is needed for eye pigment. And on the other side, we've got a promoter that's going to drive expression of that. So if we induce recombination between these, that will make a reconstructed white plus gene, and we can see that cell as being pigmented. And it doesn't matter whether that segregates as an X or as a Z, both those cells are going to be pigmented. So we can see the occurrence of exchange by the production of pigmented cells in the eye. So normally the eye of this fly is going to be white because over on the X chromosome it carries a white null mutation. But if we make this happen early in development, it'll make this cell, that cell will proliferate and make a clone that's pigmented in the adult eye. So that's how we're going to see recombination happen. And then we have to have a way to detect how those chromatids segregated. Did they do X segregation or Z segregation? And all we have to do that is put a marker out here. In this case, it's the recessive eye color mutation scarlet. And now, if we do X segregation and we make a white plus cell, so it's got pigment, will it be homozygous for this other eye color mutation scarlet? Or, in which case, that means it did X segregation. Or will it be heterozygous, in other words, wild type in phenotype? And that means it did either Z segregation or G1 recombination. If you go back and look at this paper, you'll see I'm cheating a little bit. I'm simplifying this for the sake of time and for the sake of discussion, but this is essentially the idea. All right, and I said if you can make a heterozygous cell by either Z segregation or G1 recombination. Just let me point that out to you. This is recombination in G2. Z segregation gives you a scarlet over plus cell, meaning both these center chromatids went together. Recombination in G1 gives you exactly the same thing, gives you a scarlet over plus cell. So we can't tell those apart from this particular experiment. But we did that experiment, and we found out that X segregants accounted for about 26%, Z segregants about 12%. So we're right at the same point Pimpinelli and Ripoll were, where we see about X segregation outweighs Z segregation by about 2 to 1. Now, in order to figure out if any of that is actually G1, we can throw a little twist into this experiment, and that is to throw another FRT right here. All right? And what that other FRT is going to do is allow sister chromatid exchange to occur within this chromosome, and that's going to reverse the pattern of segregation. All right, so now X segregation will still have this centromere going up and this centromere going down, but when this centromere goes down, it will now be accompanied by this. All right, so basically what we're doing is we're turning what was Z segregation into something that looks like X. We're turning what was X into something that looks like Z. So now this is going to result, this heterozygote is going to result from X segregation or G1 recombination. This is going to result from Z segregation, and now that's going to allow us to determine how much of that Z or G1 is actually happening. And the answer is Z segregation virtually never happens. Okay? So when you do this, this is all you see. You virtually never see this phenotype. All right? So that told us that, in fact, that 2-to-1 ratio is actually 2/3 of the time recombination occurs in G2 and is always followed by X segregation. 1/3 of the time recombination is happening in G1. There's virtually never Z segregation. All right, so I went over those 2 points. But then the next point is, how is X segregation achieved? So if X segregation is what happens all the time, what's the basis for that? All right, I'm going to skip over a bunch of experiments that I think Larry would have really liked, but the point of them is simply to show that experiment was done in the soma, looking at eyes. We could also do an experiment that looks at how chromosomes segregate in the germline, and the point of that was that X outweighed Z or G1 by about 3-to-1. All right, it was pretty similar to what was happening in somatic cells. All right, here is a very similar but different experiment, and now this is an experiment that we're doing in the male germline. All right, and what we're doing here is we are using FLIP to catalyze an exchange between an FRT inserted way out at the tip of the X chromosome and another one inserted way out at the tip of the left arm of chromosome 3, and that's going to make a translocation, and the bits that are exchanged are very tiny. All right, so in fact, hypoploids are going to survive here. And if we have, and I should point out, just so you're not confused, what I'm showing here are the sister chromatids of the X and the sister chromatids of chromosome 3, but I'm not showing the homologs at the top, right? But actually, these exist in a diploid cell where they exist with their homologs, and now this is after segregation where I am showing the homologs. So if we have X segregation, we're only going to follow this guy, which is going to be white plus, right? Promoter driving the 3' end of white. So we're going to be able to follow the cells that have white plus, and if they did X segregation here, they will have this half of the translocation, the other half of the translocation segregated away, right? If they do Z segregation, they will have both halves of the translocation. That means both of these guys go together. Here's one half, the half that's marked white plus. Here's the other half. And if this cell with both halves of the translocation goes through meiosis, produces gametes, when the X's and the 3's segregate, they will show independent assortment. Half the time, the translocation halves will segregate together. So that's going to allow us to determine how often this happens. All right? When we have exchange out here very near the chromosome tips, do we ever see Z segregation? Do we see G1 recombination? And the answer is that about half the time we get things that look like they did Z segregation, or they could have done G1 recombination. Okay? Now, we repeated this experiment with FRTs inserted here, which should reverse everything, and the result of all that was that this ratio didn't change, and what that tells us is that there is no preference for X segregation or Z segregation when you have exchange out near the tips, all right? Because what we're actually seeing is that X and Z segregation are essentially equal, and in this case, there's almost no G1 recombination. Okay, so again, the trick was just to put an extra FRT here, turn X into Z and Z into X. So the result's the same. It says X and Z are equal. All right, how does that get us to mechanism? Well, if you think about exchange in meiosis, this is what we think of as a nice meiotic bivalent, where the homologs are indicated by thick or thin lines, and one of those chromosomes has, or both of those chromosomes have exchanged with each other. So here is one recombinant where we've got a thin hooked up to a thick. Here's the reciprocal recombinant, thin hooked up to thick. And this now is the kind of structure that we're going to see in meiosis when homologous chromosomes have undergone a single exchange. And what is it that makes this bivalent? By the way, this is pronounced bivalent, not bivalent. What are we, chemists? So this bivalent is held together. It's created by exchange between homologs, but it's held together by sister chromatid cohesion between these arms distal to the site of exchange. All right? So it's this cohesion and this cohesion that's holding this chromosome and this chromosome together in a bivalent. Now, what we have learned primarily from work from Scott's students like John Merriam and especially Scott Hawley is that if the amount of sister chromatid exchange between these two arms distal to where you had the crossover is minimal, those chromosomes will fall apart. Okay, so that's why in that experiment where we had exchange very near the tips, there was no preferential X or Z segregation. So looking back at this, if you don't believe that's what a meiotic bivalent looks like, here's a picture of bivalents from mouse myocytes in metaphase, and I've just pointed out a bunch of them that have single crossovers, and they form this nice cruciform structure. That one actually might be a double crossover, but they form these nice cruciform structures just like that cartoon that I just showed you. So how do we explain preferential X segregation in mitosis? It turns out if you look at the chromosomes after you've induced exchange, you see exactly these same kinds of bivalents. All right? So an exchange in mitosis in combination with a sufficient amount of sister chromatid cohesion distal to the site of exchange will produce a bivalent, and that can direct segregation. So here's our bivalent where I've marked parental chromatids and recombinant chromatids, and now this wants to attach to the mitotic spindle, and because of this rigid structure, it will direct segregation of one recombinant to the right and the other recombinant to the left. So we know not only does exchange in G2 is always followed by X segregation unless the exchange is near the tips, but we know why. Because it makes a bivalent. All right, I pretty much went over all that. So a second thing that I want to talk about is, and I'm going to try and get through it quickly, is, and because I think this would be especially nice for Larry to hear, and I would be proud to be able to tell him, is a story about dicentric chromosomes, their breakage and healing, and again, we're going to use site-specific recombination, and we discovered that if you have inverted FRTs, that a very frequent recombination event would be an exchange between an FRT pointing one direction on one chromatid and the other direction on the other chromatid, and that will make a dicentric chromosome and an acentric chromosome fragment. That's a very efficient process. We can make that happen in close to 100% of the cells. This is just a nice picture of it where here is a control karyotype. Here's a normal 3. This is actually TM6. That's why the centromere is not centered. And here's a dicentric 3, and here's the acentric fragment made by that kind of recombination that I just showed you. All right, so we frequently do this kind of experiment with the Y chromosome. We use a Y that we call DCY. It's marked with a dominant marker on one end and a dominant marker on the other end, and the only point of all that is that when you make a dicentric, one dominant marker is lost to the acentric. When this goes through mitosis, it breaks, and these broken ends can be healed by de novo telomere addition. All right? And if you do this in the male germline, you can carry out a test cross and see the transmission of a chromosome like this, because now it will have this dominant yellow plus, but it will have lost this dominant bar stone, and it's recovered in a normal, viable offspring. So we can see breakage and healing in the germline by this kind of experiment. And this is just one circumstance in which we might do this. So here we have this flip. We're expressing flip by heat shock, and this is that dicentric forming Y, and we'll mate him to just a control female. And if we look at all the progeny that come out, about 90% of the sons have the intact Y. but a bunch of them have this broken and healed Y. So in this particular experiment, about 11% of the Y chromosomes that are transmitted by this male have broken and then had a new telomere added to their end. And then, of course, we've got the daughters, which are present in about the same frequency as the sons. Now, this experiment becomes really interesting if you do it in a background where you have disrupted the DNA damage response. So this is a very simplified view of the DNA damage response, but we imagine up at the top end we have ATM recognizing this in concert with some other proteins. It phosphorylates CHEK2. CHEK2 phosphorylates P53. P53 is a transcriptional regulator. It turns on a whole bunch of things. The really fun experiment for us was when we broke this process by mutating CHEK2 in particular is the most interesting one. And when you do that, you find out, first of all, that you get a lot more of the broken and healed Y chromosomes. So instead of 11% of the Y chromosomes being broken and healed, 90% of them are broken and healed. The other really interesting thing is that we have a lot more daughters than we do sons. So we are losing about half of the Y chromosomes in this experiment, all right? And we wanted to know, where are they going? What's happening here? Well, it turns out it's not zygotic death. These males are not transmitting chromosomes without telomeres that are causing death of the offspring. It's really much more interesting and fun than that. This is basically how we perform the experiment. We induce flip here. We're making dicentrics in germline stem cells. The dicentrics are formed and they break, and we've got gonial blasts with broken chromosomes. In a wild-type male, what happens is mostly those cells die right away. A few cells escape because they get a telomere added before the cell has a chance to die. So mostly what you have are cells that haven't experienced dicentric formation, going through 4 mitotic divisions, 2 meiotic divisions, making a bundle of 64 sperm heads here in this bundle of elongating sperm. All right? But in a CHEK2 mutant, you make the dicentric, it breaks, the cell doesn't care, it goes on and divides again, and during those 4 mitotic divisions, it's doing what Barbara McClintock first discovered, these breakage fusion bridge cycles. So we make an anaphase bridge, it breaks, the cell doesn't care if it doesn't have a functional CHEK2 checkpoint. It goes through S phase, it still knows that there's a problem, all right? And it wants to repair the ends, but it's not going to stop the cell cycle to do it. But it fuses those ends, it goes through mitosis again, and this whole thing repeats and repeats until you get to meiosis. How do we know that's happening? We looked. So we sort of violated one of Larry's tenets that you don't really need to use the microscope to understand how things are working. It's better to just do crosses, but we would not have figured this out through crosses, all right? And when you look at these sperm or these testes and these groups of, in this case, cells going through meiosis II, you see all these chromosome bridges. So these are a result of an event that happened days and days ago in the germline stem cell, all right? But now that chromosome has not been healed. It's been doing these breakage fusion bridge cycles until finally it gets to meiosis II. Meiosis II, because these are sister chromatids that are fusing, right? And these are all Y chromosome bridges that we're seeing in meiosis II. And you can see them even in cells where cytokinesis is essentially complete. Now, cytokinesis doesn't really complete at this point. But these bridges are not breaking at this point. They've been breaking through all the mitotic divisions. They don't break here. What happens is, as the sperm mature, as I said, they mature in a bundle where normally you have 64 sperm heads up here, but now in this background where we induced dicentric formation days ago, these differentiate and defective sperm heads are being swept away down the length of the sperm bundle. They will never make mature sperm. If you look closely, you can see things that look like broken bridges, or you can even still see bridges connecting one sperm head to another sperm head. All right, so we know exactly why we have that sex ratio distortion. why we're recovering X chromosome sperm, but not Y-bearing sperm. It's because the Y-bearing sperm are being eliminated because they had a bridge in meiosis II. Why do I think Larry would have liked this? So I think it's been mentioned that Larry, I think Barbara mentioned that Larry is widely known for a lot of things, but one of them is for the discovery of segregation distorter. And this is a slide from Larry, Yuichiro Hirazumi, and Iris's paper on SD, where they actually, the very first paper on SD, where they proposed a model for how segregation could occur. And what they proposed was that the SD chromosome makes a break in the SD plus chromosome in the heterochromatin, that those two ends, those two broken ends, then fuse, they make a bridge in meiosis II, and that turns out to be lethal to sperm. So we think this is probably not how SD works, but I think it's really interesting that Larry predicted exactly what would happen if you did have a bridge in meiosis II, right? It's going to make a bridge, it's going to be lethal to sperm, the homologs are fine, they go on and produce mature sperm. So I just want to finish with one other comment, and that is that although we don't have Larry around anymore, one thing that I really do appreciate is the community of Larry's students. Bill and I were students together. Gary Carpin was a student in Charles's lab at the same time, and I know that I had many experiences with Bill in the lab that I cannot forget, no matter how hard I try. And even people who weren't students with us, Barry, Scott, you know, I still feel like we're part of that community, and it's always a pleasure to see them at meetings and hear about the interesting things they're doing. And it was a real pleasure to be a student in Larry's lab, and I thank him for that.